this very kind introduction, Josep. So yeah, Josep said, and as you can infer for the title, I will be speaking about music and the neural mechanisms supporting music-induced pleasure. So people generally rank music as one of the most pleasant experiences in their life, and it has, without doubt, a strong motivational impact on us. And one example that I generally like to use in my presentations that I think that it nicely reflects the music's power is the existence of festivals like yeah, okay, now the existence of festivals like the Igloo Fest. This festival is held at Montreal in January, outdoors. So for those who are not familiar with the weather in Montreal, in January, we may reach very easily like minus 20 degrees or so. So it's very, very cold. Actually, everything in this festival is built on ice, the amenities, the bar, everything. And I think its existence is a very nice example of how people are willing to spend their money, because it's not a cheap festival, to spend their time as well. Not only the time that they spend in the festival listening to music, but also sometimes there are huge lines to have access into the festival. And these lines, you're waiting there at minus 10, minus 20 very easily. And also how people are willing to sacrifice their own comfort, right? Because they could just stay at home watching Netflix under a blanket, but they just decide to go there, right? So that's something that really intrigues me. It's to understand what's going on in our brain, how our brain translates a structured sequence of sounds such as music into a pleasurable reaction and a reward. So one of the first studies that investigate this topic was this research done by Blood and Satorre in 2001. So basically, what they wanted to do is they wanted to investigate which brain regions were associated with this experience of pleasure with music. So to do so, they asked participants to bring their own favorite music into the lab, and they specifically asked for music inducing chills or goosebumps. Basically, because chills and goosebumps are clear, discrete, and highly reproducible events that are generally experienced with highly pleasant music. And for that reason, chills are generally used as an index of the degree of pleasure that participants are experiencing. Besides listening to their own favorite music, they also listen to a control music, which consists basically of, so basically each participant listened to their own favorite music as well as other participants chill-inducing music that did not elicit chills on them. Okay, so other participant favorite music that for a particular participant was not really pleasant at all. So that way, the participants ensure, the researchers ensure that any potential differences between chill-inducing music and control music was driven by the subjective experience of the participants rather than some common acoustic features that could share all these chill-inducing music. And basically what they did is they measured physiological responses as well as brain activations while participants were listening to these two different type of conditions. So at the physiological level, they basically show that when listening to chill-inducing music, there was an increase in individuals' physiological responses, heart rate, respiration, skin conductance, and so on, as compared to when participants were listening to the control music, indicated that this physiological response could be actually good objective measures of the experienced pleasure by the participants. And in terms of brain activity, what they found was that chill-inducing music was actually associated with a distributed network involving both cortical and subcortical regions that were previously involved in motor control, such as SMA, putamen, and cerebellum, emotion, such as anterior cingulate cortex, insula, or amygdala, and more importantly for us, also involved in reward processing as the ventromedial prefrontal cortex, and more important for us today, the ventral striatum, including the nucleus accumbens. So this nucleus accumbens is a key hub of the reward circuitry. It plays a key role in different aspects of reward processing, from reinforcement learning to motivation to pleasure. And it's a structure that it's preserved across evolution. So it's a structure that is quite phylogenetically old. Okay, that's something that we share with many other organisms. And actually that we generally find activations of this structure when participants are receiving monetary rewards, food rewards, or erotic rewards. So this first study of Blood and Satorre already point out that despite music complexity, sophistication, and abstractness, at the end it may rely in an otherwise quite ancient and phylogenetically old circuitry as it is the reward circuitry. So another evidence supporting the role of this reward circuitry in musical rewards come from an investigation done by Salimpur and colleagues 10 years later in 2011. And here what the researchers wanted to test was to investigate whether music could lead to a release of dopamine, a well-known neurotransmitter for its role in reward processing. We know that it controls, or the different regions of the reward circuitry are somehow under control of dopaminergic transmission, and also dopamine is the responsible of the addictive properties of sweetness, for instance, but also of drugs of abuse, which generally cause a big, fast increase of dopamine in the brain. So basically, the question was, would then dopamine also mediate the rewarding properties of music? And to test this hypothesis, what they did is they took advantage of PET to not invasively measure dopamine release in a group of healthy participants. And the design was exactly the same than the previous one in Blood and Satorre 2001. So again, participants were asked to bring their own favorite music that would induce them chills and goosebumps and so on. And then as a control, each participant listened to another participant chill-inducing music that did not induce chills on them. Okay, and they also measure physiological response, and they extend the findings of Blood and Satorre, showing that these physiological measures can not only discriminate between chill-inducing music and controlled music or neutral music, but it's also sensitive to the intensity of the chills. So again, pointing out that these physiological responses might be a good objective measure of musical pleasure in this context. But the most interesting results that they found was that chill-inducing music was actually associated with dopaminergic release in two key structures within the striatum, in the caudate and in the nucleus accumbens. More importantly, release of dopamine in these structures was correlated with participant reports of pleasure, in a way that dopaminergic release in the caudate predicted the number of chills that participants experienced during the session, and on the other hand, dopaminergic release in the ventral striatum in the nucleus accumbens predicted not the number of chills, but how intense these chills were, and also subjective reports of pleasure that they provided at the end of each exercise. So this study, and together with the investigation of Blood and Satorre, further support the hypothesis that musical reward relies on a similar brain mechanisms than those previously identified in sensory pleasures, such as with food or drugs of abuse. However, all the evidence that we have seen so far comes from neuroimaging studies, and those, they can only provide correlational evidence. So all we can say is that when people listen to music that they like, there is engagement of the nucleus accumbens, for instance. But we cannot disentangle whether the engagement of the nucleus accumbens is actually causing that pleasure, or if it's responding only as a consequence of that pleasure. So to provide causal evidence, what we need to do is to somehow modulate the functioning of the circuitries and see whether that actually turns into changes in pleasure and motivation in music. So that's something that it can be easily done, or is a gold standard in rodents. So they can be super precise with very precise surgeries, targeting very specific brain regions. But with humans, of course, this is not that easy, right? So yeah, that's a challenge. However, nowadays we do have techniques that allow to non-invasively stimulate the human brain. One of them is the transcranial magnetic stimulation. However, the main limitation of this technique is that it has a limited range of action. We may only stimulate cortical regions, those that are more located in the surface, but there is no way that with TMS you can directly stimulate the striatum. It's too deep, this brain structure, to stimulate with TMS. However, one thing that we have to consider is that the striatum isn't, as any other part of the brain, is not working in isolation, but in interaction with many other brain regions, and particularly with cortical regions. And what some people have seen, and one of the first studies was done by Strafella in 2001, is that because these functional and anatomical connections between certain parts of the cortex and the striatum, it is possible to indirectly modulate the functioning of the striatum by just stimulating these specific cortical regions. And in particular, what the Strafella found was that stimulating with TMS the left DLPFC would lead to a release of dopamine in the striatum. Although we were not directly stimulating the striatum, but we were somehow stimulating the circuit, and as a consequence, what they saw was an increase of dopaminergic release in striatal structure. These findings have been replicated in humans and also in rodents. Also, fMRI studies have shown that this procedure can modulate striatal activations. So it seems like a good way, at least to start, something to start with, to see whether the striatum do play a key role in musical pleasure. So basically what we did, what we were willing to do, was to take advantage of this method to modulate striatal function and see whether that would cause changes in pleasure, in the number of children that they were reporting, but also in their motivation to purchase music or to listen to music again. So with this goal in mind, what we did was a study, first a behavioral study in 17 participants, which came into the lab in 3 separate sessions. On each session, they would receive either inhibitory stimulation, or the protocol is called CTBS, continuous theta burst stimulation. In another session, they would receive the excitatory stimulation, which is called intermittent theta burst stimulation. So important here, the I doesn't mean inhibitory, but intermittent, and it's excitatory, just to clear things up a bit. And a third day, they could receive also a sham condition, in which basically we would use either one of these 2 protocols in counterbalance order, counterbalance across participants, but what we would do would be just flip the coil just before the stimulation would start, so that participants' brain would not be stimulated, just the room's air. Okay, these protocols are very fast. It takes like CTBS, it lasts around 40 seconds, super fast, and the excitatory stimulation lasts 3 minutes. So the idea is that the effects of this stimulation may last. It's difficult to assess for how long it may last, particularly when you are stimulating prefrontal brain regions. But what is thought is that these effects may last for about 30 minutes to 1 hour. Okay, so when participants came into the lab, we explained them all the procedure. We stimulate them with one of these 3 different protocols, and after that, we sit them in front of a computer with the headphones, where they would be listening to music, and no more stimulation happened. Okay, we were just analyzing what would happen when they were under the effects of these 3 different types of stimulation. And basically what they did, they just listened to music, in particular 15 musical excerpts, and that include their 5 favorite songs, similar to what Blood and Sat already did, and also they listened to 10 songs that we selected specifically for this experiment, and these 10 songs were different from session to session, and you will see now why we did that. And during the whole session, we were acquiring skin conductance response, an objective measure of emotional arousal, and as we previously have seen, highly associated with musical pleasure in this type of task. So what participants had to do was basically to indicate the degree of pleasure that they were experiencing while listening to the music. They did that by pressing any of 4 buttons that they had available in a keyboard, one indicating that they were not experiencing pleasure at all, and the maximum rating was the 4th button, which would indicate that they were experiencing a chill or a goosebump or something like that. And they were doing that in real time. And a part of that, they also had the opportunity to purchase our music selection. So they had the possibility to bet, after each excerpt, indicating how much money they were willing to pay to buy that song. And that's why these 10 songs were different from one session to another, to make sure that in every session they were betting for completely new musical excerpts. So basically, we use these subjective reports of pleasure and these physiological measures as an index of the experienced pleasure. And we use the amount of money that they were willing to pay as an index of participants' motivation to listen to music again. So it's quite simple design. We only have 3 outcome variables. And basically what we did here, we use sham as our baseline. So for each of these 2 sessions, we compute percentage of change in any of these 3 variables following inhibitory or excitatory stimulation as compared to sham, and then see the differences between the 2. So in terms of subjective reports of pleasure, we found a main effect of stimulation. So basically, following excitatory stimulation, participants, there were positive changes following excitatory stimulation. So people were more likely to enjoy the music than at sham. And on the contrary, following inhibitory stimulation, what we saw was negative changes. So people were less likely to enjoy the music. And this was true for their own favorite music, but also for our music selection. So there was no condition effect in there. And kind of the same apply for the objective measures of pleasure in terms of the amplitude of the electrodermal activity while listening to pleasure music. And again, positive changes following excitatory stimulation, negative changes following inhibitory stimulation. And again, we saw a main effect of TMS stimulation in the amount of money that participants were willing to pay. Again, positive changes following excitatory stimulation, negative changes following inhibitory stimulation. So our sort of conclusions were that these findings were supporting the idea that striatal function causally mediates musical reward processes, either the hedonic reactions as motivational aspects of reward. However, a limitation of our study is that we didn't measure any brain activity. So we were assuming that these effects were basically driven by some modulations in the striatum, but we didn't measure what was going on in our participants' striatum. So it could be perfectly possible that these effects were merely driven by changes in another brain structure, even within the reward circuitry, such as the BMPFC or other striatal subregions, or it could be just driven by local changes in the DLPFC, which is actually the brain region that we are directly stimulating. So to try to disentangle a bit that and go into more details of which were the brain regions that were actually modulating this pleasure and motivational responses, we performed a second experiment in which participants did exactly the same. It's the same task schema, same task design, but now participants did the task in the MRI. Okay, so first, the behavioral results. So we found, as in our previous TMS study, that following excitatory stimulations, there were positive changes in subjective reports of pleasure, amount of money participants were willing to pay, number of chills, and time reporting chills. While following inhibitory stimulation, it was sort of the opposite. No, there were negative changes for each of these variables. So that was very nice. It was sort of a relief for us to see that we could replicate the same findings than in our previous study, but now with a new sample and also in a more noisy environment as it is the MRI. Next, we look at potential changes in brain activation within the reward circuit because of this TMS modulation. We look at brain activations in different regions of the reward circuitry. We basically focus in the two main structures previously involved in reward and musical reward in particular, which are the striatum and the ventromedial prefrontal cortex. And we look at different parts of the striatum, the nucleus accumbens, of course, but also the caudate and the putamen. We then analyze two different time points. I'm not gonna enter into the details of why we did that, but basically we look at brain activations at the moment that people were reporting a change in pleasure. So at the time that they were saying, look, I'm experiencing now a chill, but also before that button press. So the time that we call like some sort of anticipation phase. And we look at these two different time points basically because the reward circuit has been involved in reward experiences, but also, and almost more importantly, in the anticipation of rewards. So no matter which window we look at it, we found a main effect of a stimulation in all these brain structures. So here the 0 represents the sham, sort of saying. So positive changes mean greater activations than in sham. Negative means lower activations than in shams, following either excitatory stimulation or inhibitory stimulation. As you can see, generally what we found was positive changes following excitatory stimulation in these four structures and negative changes in these four structures following inhibitory stimulation. We check also whether the effect was present in other control areas. We didn't see anything. We didn't see anything, either an effect in the DLPFC, which is something that at first might surprise you because that's the brain region that we are targeting. But it's important to take into account that here the contrast was not addressed, but was what we were looking here is brain activation as a function of musical reward. So the contrast was like comparing highly pleasant music versus non-pleasant music. And in this contrast, generally, we don't see anything. We don't see the DLPFC involved. Actually, most of the studies that have investigated the neural basis of music-induced pleasure, none of them have found a crucial role of the DLPFC. So basically, the DLPFC is not modulated by music reward. And what we see is that they remain like this, independently of whether we stimulate this DLPFC more or less. We would probably see changes in the DLPFC if we would analyze brain activations at rest immediately after the stimulation. But when we look how this brain region, how brain activity changed as a function of music reward, we don't see an effect of the DLPFC there, only in these reward-related structures. That was nice. That means that somehow our procedure was working and there were differences in the activation of this reward circuitry, depending on whether we were exciting or inhibiting the circuitry. However, what we wanted to know is which of these brain regions were actually behind these behavioral effects. So which TMS-induced changes in these different four structures could account for TMS-induced changes in these patterns of behavior in relation to pleasure and motivation. And among these four different brain structures, only the nucleus accumbens seem to predict, so only TMS-induced changes in the nucleus accumbens predicted TMS-induced changes in subjective pleasure and motivation, although it did sort of dissociation. So basically, TMS-induced changes in the nucleus accumbens during the experience of musical pleasure were associated with TMS-induced changes in subjective reports of pleasure, the more the better. And this effect was not present when during the anticipation phase, But instead, in the anticipation, what we found was a correlation between TMS-induced changes in the nucleus accumbens during this specific phase and participants' motivation to buy our music selection. So the amount of money that participants were willing to pay. Again, the greater the TMS-induced changes in the nucleus accumbens during the anticipation, the more money participants were willing to pay to buy our music selection. So overall, these findings indicate that the engagement of striatal dopaminergic pathways and the NAC in particular is indispensable to experience rewarding feelings of music. Yet this study had another limit of our we're talking about dopaminergic striatal pathways, basically because we see that the changes are in the NAC. We know that the nucleus accumbens is one of the main target of dopaminergic neurons. Previous studies have shown that the procedure that we are using here modulate dopaminergic release in these structures. But of course, we were not measuring dopamine here, and everything that we were doing was a very indirect brain stimulation procedure, which could perfectly stimulate or modulate other neurotransmitters. So we couldn't really go very deep into the neurochemistry behind these effects. And to try to solve that, what we did was a third experiment in which now what we did was to directly modulate dopaminergic levels. So what we did here, we tested like, I'm not wrong, 27 participants in a pharmacological intervention. Again, the task schema is exactly the same than the other two previous studies, but now we just changed TMS for drugs. One that was levodopa, a well-known dopaminergic precursor. Then they could also get a placebo, which was just a pill full of lactose. Otherwise, they could get risperidone, which is a dopaminergic antagonist. So same as in the other two previous studies, participants would come sessions and they would receive either one or the other in a counterbalance order. Okay, so this is it. And what we found here was sort of the same that what we found with the TMS experiments. So basically here the colors change. Green is levodopa, which is a dopaminergic precursor, and red is risperidone, which was the dopaminergic antagonist. So basically what we saw was that following levodopa administration, participants were more likely to experience chills. They also present higher amplitude in their skin conductance response or electrodermal activity, and they were also more willing to spend money to buy our music selection. While with risperidone, it was the other way around. People were less likely to experience chills, lower skin conductance response, and they were less likely to spend money on our music selection. So this series of studies indicate that music-induced pleasure do rely indeed in an otherwise evolutionary ancient circuitry through dopaminergic control. These findings, however, which might seem consistent with previous animal studies, they are not somehow. Basically because all other previous studies that have basically in rodents using sweetness as a primary pleasure, what they have found is that dopamine doesn't really modulate hedonic reactions to food or to sweetness, but it rather change or modulate participants' motivation, animals' motivation to work for a particular reward. So if you modulate dopaminergic levels in a rat, you will not see any changes in terms of their affective reactions to sweet, but what you will see is that they start pressing a lever as crazy to get that food, but nothing else. So that somehow points to a probable dissociation between high-order pleasures, in which dopamine seems to play a causal role, and more primary pleasures, in which dopamine doesn't really play a role in terms of affective reactions, but it's more specifically focused on motivation and learning. And to be honest, when I started this series of experiments, what I was expecting was that we would maybe modulate the amount of money that participants were willing to pace so that we would modulate participants' motivation. But that my hypothesis was that probably we wouldn't see anything in terms of subjective reports of pleasure or physiological responses associated to pleasure. Yet we saw that not only in one experiment, but in 3 different experiments. So how we interpret this inconsistencies between our investigations and research on primary pleasures. So basically what we think that it's going on here is that musical pleasure, in contrast to what occurs with more primary rewards, may rely on cognitive operations, in different cognitive operations, such as expectations and surprises driven by the structural and temporal regularities present in music, or on associative learning or episodic memory, among others. And most of them are cognitive computations that rely on dopaminergic transmission, from dopamine's role in learning to its role in memory, working memory, or attention. So one potential interpretation of our results is that dopamine-dependent cognitive processes, such as learning and memory and so on, may substantially influence the pleasure and the way we appreciate abstract activities as music. However, we may wonder, okay, that's very cool, but if everything at the end relies on an ancient circuitry that we share with many other organisms, why it is that it seems like music and our appreciation for more aesthetic experience seems to be quite a human trait. You know, if monkeys also have this hardware, why we have not been able to see a preference for music in non-human primates? Well, uh, in here it's important to, so in order to assess if there are some specific reward circuitries involved in music as compared to the processing of primary pleasures, such as food, or secondary rewards as money, we try to investigate this question using an individual differences approach. So basically what we tried to see was to identify people who do not like music, but do like other stuff and have no problems with music perception. So people that can fully understand music, that can experience pleasure with other type of activities that it's not music, but with music they don't really care at all. So when we started this project, that was the idea. Let's find these people, and compare what's going on in their brain as compared to other people that do enjoy music. And that may help us to disentangle if there are or there are not specific reward circuits involved in music reward. So first thing that I did was to Google it, if there was somebody somewhere saying that they had this sort of phenotype. And I found at first like a couple or 3 of people around the world reporting exactly that, that they didn't really care that much about music, that it's not that they dislike, it's just basically that they didn't care, although they were perfectly able to appreciate other things like pictorial art, books, food, and so on. So in order to see whether we could identify this population and so on, what we did first was to develop a questionnaire to assess individual differences in music reward sensitivity. And that's a questionnaire that if you go to our Brainvitge website, you can check which is your music reward sensitivity. It's a very simple questionnaire with only 20 items, and it gives you an overall score as well as other scores more specific for the different facets that we identify while developing this questionnaire. So while we were developing this questionnaire, because we knew from previous studies that music and other type of rewards, they all seem to engage and to have in common this reward circuitry, we also administrate other questionnaires related to more general reward processing and hedonia in general, just to somehow validate our question. We would expect some sort of correlation, and actually we did. So musical reward sensitivity was correlated with different measures of the BISBAS questionnaire, as well as with the physical anhedonia scale, which makes completely sense. So people that are more sensitive in reward processing in general, they are also more likely to be very sensitive to music reward in particular. However, there were a group of participants that were not exactly following this rule. And actually, we found that around 5% of the two samples that we analyzed presented average scores in these more general reward processing questionnaires, although they still present pretty low scores in the BMRQ. So they seem to be people as those that we were looking for. So people that did not really like music or that didn't really care about music, but still were able to experience pleasure in as average people as or in normal with other type of rewards. However, here everything were just self-reports, so it could be perfectly possible that I don't know that this participant just got confused answering the questionnaires and they did it the other way around. That also another possibility was that these individuals were just having some difficulties with music perception, and actually there is a condition called amusia, and amusic individuals generally have deficits in music perception, particularly in pitch processing, and they generally are less likely to experience pleasure with music because they don't get anything. So in order to disentangle a bit all of this and try to disentangle all these different possibilities, we then perform a second experiment in which we select three groups of individuals with low sensitivity to music, average sensitivity to music, or high sensitivity to music based on the BMRQ, but that they present similar, but with similar sensitivity to other kind of rewards, to food, money, sex, exercise, or drugs. And to do that, we use several questionnaires, and we make sure that the three groups were matched according to according to different questionnaires of reward sensitivity. And also, we made sure that these three groups did not differ in their music perceptual abilities, so that there were no amusics in here that could somehow explain the correlation that we saw before. So we asked participants to come into the lab to bring their own favorite music. And because we were afraid that maybe anhedonic individuals would have some difficulties in selecting highly pleasant music, because by definition, we would expect them to be not very sensitive to music, we also create a music list of highly pleasant music based on a survey that we did in almost 200 students of similar age and similar socioeconomic status and so on. And actually, our fears come true. So anhedonic individuals had difficulties to bring their three favorite pieces of music. One of them just brought one, another one brought two, two more said that they had a lot of difficulties, that they need the help of her sister or their brothers or their friends. And basically what they were reporting is that for them was very difficult, basically because they had no strong preference for any type of music. And that's something that we didn't see in any of the participants included in the hedonic and hyperhedonic groups. There were 10 people on each group. So they came into the lab, we make them listen to their own favorite music and also our music selection, and again, ask them to indicate the degree of pleasure that they were experiencing, reporting chills, the intensity of these chills, and we also ask them to provide a more general pleasantness rating by the end of each exam. And what we found is that behaviorally, there was a very nice correlation between the BMRQ and the subjective reports of pleasure reported while listening to the music, also in terms of the intensity of the chills that they experience, and also in this more overall assessment of pleasure that they provided at the end of each exam. In addition, we also measured physiological responses and while they were doing the task. And here you can see the skin conductance response when participants were reporting to experience no pleasure, low pleasure, high pleasure, or a chill. In red, you can find the skin conductance response of the anhedonics. In green, the hedonics, or the average sensitivity to music group. And in blue, the hyperhedonic, so those that really get crazy with music. And as you can see, the skin conductance response of the anhedonics is sort of flat. It doesn't seem to be super modulated by musical pleasure. Here it is slightly, but that was actually driven by only one single participant. But in the other two groups, we see how this skin conductance response increase proportionally to the degree of pleasure that they are experiencing, as we would expect based on previous evidence. And in particularly in the chill, we can see huge differences between the anhedonics and the hyperhedonics and hedonics. All the hedonics and hyperhedonics participants, on average, after a chill, you would see an increase of a skin conductance response, something that the few other hedonics that reported chill didn't really show. So that was nice, but that could be, you could say, well, that's just, that may be driven because the anhedonics, they have a general deficit in their skin conductance response. It doesn't matter which task they will do, that you will always see this low increase in their skin conductance response. So in order to control for that, participants also did a second task, which was a monetary reward task, which they could either win or avoid losing money. And what we saw is that first, behaviorally, the three groups were very comparable. They won similar amount of money, and their reaction times were very similar, but also their physiological response were very comparable. We didn't see any group effect in any of these measures, neither behaviorally or physiologically, in the monetary reward task. So so basically here in this study, what our conclusion was like, look, these people exist. There are people that do not like music, that do like other stuff and have no perceptual problems. And these people might be a great model to actually understand which are the specific brain circuits involved in musical pleasure. So for that reason, we performed a second study, again with 3 separate groups of participants, as we did before, individuals with low sensitivity, average sensitivity, and high sensitivity to music, but with similar general sensitivity to reward and similar music perceptual abilities. And here what you can see is brain activations during the monetary reward task in the nucleus accumbens, because that's the particular brain region that we are interested in. And we didn't find any group effect in this task. So in the 3 groups, you could see the expected increase of activation in the NAC following monetary rewards respect to losses, for instance. And however, when participants were doing the music task, we didn't see this pattern. We actually saw a very similar pattern that what we found previously with our physiological data. So basically, there was increase of activations in the nucleus accumbens associated to music reward in the hedonics and hyperhedonics group, but we didn't see that in the anhedonic group. So it seems, so these findings somehow extend our previous findings, suggesting that this lack of sensitivity to music in musical anhedonics is basically driven by a specific lack of response in the nucleus accumbens to music. But why is that? So another interesting result here is that the 3 groups did not only differ in their brain activations in the nucleus accumbens, but also in the communication between these nucleus accumbens and the right STG, where the auditory cortex is located. So basically what we found is that where there was connectivity between these 2 brain structures, these 2 systems, as a function of musical reward in the hedonic and hyperhedonic group, we didn't see that in the anhedonic group. So something that could explain this specific lack of response in the NAC in response to music could be that this specific musical anhedonics, the problem that they have is that they have a low connectivity between this high order cortical structure involved in auditory perception and cognition, and on the one hand, and the reward circuitry and the nucleus accumbens on the other. So in a second study, we also investigate whether this musical anhedonia condition was associated with differences in white matter tracks. So for that, we acquired DTI data. The data was coming actually from the same participants of the fMRI study. And what we found was that white matter paths that were connecting the superior temporal gyrus, and the nucleus accumbens through the orbitofrontal cortex were mediating the role of, were mediating and were explaining as well this music reward sensitivity, and were mediating the relationship between brain activations in the neck and music reward sensitivity. So, and why through the orbitofrontal cortex? Well, basically because there is no direct anatomical connections between these two. So we thought that maybe that was driven, this functional connectivity was driven by a third brain region, and the orbitofrontal cortex is actually a great potential candidate because receive inputs from auditory cortex and send them back to the accumbens. And that's what we found in terms of white matter. So these findings suggest that a specific musical anhedonia present reduced communication between the auditory cortex and the reward circuitry. But it also speaks about musical pleasure, in which doesn't rely only on the functioning of this ancient reward circuitry, but also on how this ancient reward circuitry interact with other cortical regions that are somehow more evolved in humans and that are involved in auditory perception and cognition. So the two take-home message of today's presentation would be that despite the sophistication, complexity, and abstractness of music perception, music-induced pleasure relies on an ancient circuitry, this reward circuitry, through dopaminergic control, although it seems there is some sort of dissociation here between the role of dopamine in high-order pleasures and the role of dopamine in more primary pleasures, but also that in order this pleasure to occur, it's important not only the functioning of the circuitry, but how the circuitry is connected with cortical regions involved in auditory perception and cognition. And well, finally, I just want to thank all these people who made this, all this research possible.
Thank you very much, Ernest. It's been a super nice talk. So now it's time for questions. Remember that you can raise your hand with this icon that you have on the bottom of your screen, and then we can give you access to your microphone. No questions. I do have some. So I will start. Ernest?
Yeah.
Do you hear me well? Okay. I have a bit of trouble or trying to understand this separation between motivation and pleasure. You have been talking about primary pleasures in rodents, in which dopamine seems more related to motivation than hedonic responses. And I was wondering if that is the same also in humans, like in food, if they have been, if there are studies like the ones that you showed for music, but done with more primary pleasures. I don't know, I in my mind, if I like something a lot, I would feel more motivated, right, to repeat it or to buy it or something like this. So I have a little bit of trouble with this distinction.
Yeah, I mean, it's actually something that generates a lot of trouble in general, because as you said, they are super correlated, these two things. No, generally we seek what we like, but what research in rodents have shown is that although they are very correlated, they might be mediated by different neurotransmitters. So the dopamine would be more related to this seeking, to this motivation. And actually, dopaminergic studies in humans have also shown that generally manipulating the dopaminergic system doesn't really lead to subjective changes of pleasure, at least with food or other types of rewards. We are the first actually to show that dopamine may actually have some effect in hedonic reactions, but we are also the only ones that have tested more abstract rewards. Actually, we have from the same set of data, so the same participants that did the music task with the pharmacological intervention, they also did another series of tasks. One of them in which that Pablo Ripolles was working on, in which the participants had to learn some the meaning of some words. And what he saw with his data is that with dopamine, it seems that people were also more likely to enjoy when they successfully learn something than following risperidone, for instance. And
Yeah.
here the tricky thing is that, so it seems that dopamine may play a role with more abstract, aesthetic
Pleasure, but
rewards.
in general, yeah.
Yeah. And
Yeah.
but on the other hand, you have in rodent studies, what they show is that this hedonic reactions seems to rely more on opioid system. So that when you modulate the opioid system, you see that the effective reactions of the rat completely change when they are testing food, something that they don't see with dopamine. And we collect data with also opioid manipulations a couple of years ago. Now we have the paper ready and we're working on it, but we don't see the same that with dopamine. So we see that physiological responses are modulated, but participant subjective reports are not following opioid manipulation. So it's still hard to understand what's the role of each of them. Also with humans, we are very limited with what we can do because some of the effects of these neurotransmitters are very specific to certain regions, and we have no way to be set so specific with the drugs that we have so far. So yeah, I don't know
Okay, thank you.
if that helped.
Yeah. So what I understood is that the bottom line is that the more abstract kind of pleasure that you can get, more cognitive pleasure, let's say, related is mediated by dopamine. So I was thinking in my mind, like maybe almost everyone prefers like sweet food and food that tastes like nothing, but there are foodies, right? There are people who get chills also when they eat, and I was wondering maybe if that, or the appreciation of a good wine, which entails more perceptual and cognitive aspects, maybe would be also mediated by dopamine to a certain extent.
Yeah,
So
that's actually a very good point. I'm actually now thinking on a project in which we try to look at that using astronomy kind of as a high order pleasure, right?
okay,
Because generally when we talk about food and in the reward field, food is always considered as a primary reward. But we
yeah,
all know that food is not only about sugar, calories, and things like that. There is something else in there, and there is also complexity, harmony, things in there that are actually very similar to what may happen with music. So I was thinking that that would be a good model to properly assess differences between more high order pleasures that are more related to complexity and stuff like that, as compared to more innate pleasures as sweetness. Yeah.
cool, interesting. Okay, thank you. More questions?
Sorry, I did not raise my hand.
It's
Thank
okay,
you.
David, go on.
Yeah, thanks, Ernest, for this very, very nice, very interesting talk. I just have a very, very small question about this. I think it was the study, the most recent study that, not the, like in time, the one that you probably just published, where
Yeah.
you said that, so you applied this TMS, and then you just show the different activations according to excitation or inhibition in nucleus accumbens, caudate, putamen, and all that. Did you look at the whole brain? I don't know. Did you look into other regions? Did you see any other patterns in other regions that might be implicated in emotion or something, you know?
Yeah. So basically, at the end, we use this. We actually, I think that we published in Neuroball also the whole maps.
Uh-huh.
But we didn't see like if I know I don't remember. But I would bet that maybe there was something in the hippocampus or something like that. I would say maybe among all the other regions, but the main effects were located in these four
In this one.
brain structures. What we saw as well is that, I mean, when you were looking at the individual maps of each participant, and you would
Uh-huh.
put in a screen the three different sessions, you could perfectly see which one was the excitatory or the inhibitory. In this regard, it worked the TMS procedure worked very, very well. But there was a lot of variability in terms of a specific coordinate in which this effect was maximum.
Okay.
So that what I mean, that's why we end up doing an ROI analysis, because what you would see is that maybe all participants would have some TMS effect in the caudate, for instance, but at very different locations. So there was a large variability in this matter. We think that it's basically because we are just targeting one particular coordinate, and probably there are also individual differences in the connectivity between this particular coordinate and the striatal, which is something that would be also interesting to investigate.
Yeah. Okay, thanks.
Welcome.
More questions?
I may have another question. Because I'm really not. I'm not at all an expert in amusia or anhedonias, or especially in regards to music. But I think you said that people with amusia are not, so they are able, so they kind of not have emotions related to music. But for some reason, I always thought that these people are not able to, I don't know, perceive music per se, but nevertheless, they are still able to report the affective content of the music. Is that?
Yeah, they are able to report the affective content, and they are basically, I mean, you diagnose them based on their perceptual abilities, but it's also true that they are less likely to enjoy music, let's say like that. So it's not that they have difficulties in reporting whether something is sad or it's happy or something, but they may like it less. But it's also true that there are, I mean, some cases of musicophilics that are amusics. And there is one study that is very interesting of one music individual that really loved jazz music, and he actually was trained for about, I don't know how many months, and he received musical training, and even though he didn't really improve, at least in terms of his music perceptual skills, but this guy, he really liked music, although he was not, able to fully understand maybe or perceive music. It's also true that the music is very specific to pitch, so they may get pleasure out of other things than pitch or melody.
Rhythm, something. Okay, thanks. Thanks very much.
Okay. More questions? Okay. Right. So, Ernest, do you want to add anything else, or maybe Josette?
No.
Okay. Thank you.
Well, I may have one more question. Because at the beginning of your talk, you were talking about many different regions. I don't remember. So besides, I think it was dorsolateral prefrontal cortex, was it that might be projections or having bilateral projections with nucleus accumbens? But there were a few other regions. And I was also wondering why you chose among all these specifically that one.
So basically because we had, basically because there was this study of Estrafela and others showing that there was a dopamine release following stimulation in this very particular region on the left hemisphere. And also because stimulation, for instance, of the BMPFC or the orbitofrontal cortex with TMS, it's no way, it's too painful because you are stimulating here, it's very hard. So you will not find, at least I haven't found studies stimulating this particular region.
Okay, now I see that. Yeah, I was not looking into the figure. Now I see that it was anterior single. Okay, so that was probably, okay, so you had literature backing this up, and then it was the easiest. Okay, thanks.
Exactly.
Thanks a lot.
You're welcome.
Okay, so thank you, Ernest, for being here with us. Thanks again. And the rest of you will see hopefully next week, same time at 3, same place. Okay?
Yeah.
Thank you.
Bye.